Can you hear me? Yes. Thank you. So thank you so much, Luis, for this very nice presentation, because I can follow Spanish. I cannot speak it, but I can understand it. And it's really a great pleasure to be here. It's my first time in Mexico, and it's a great place for studying Atmosfera. And as Luis said, this is a kind of new topic. Not so new because we started about 10 years ago, 15 years ago, but it is mixing microbiology and atmospheric sciences, so it's a bit unusual. So first of all, maybe you wonder where do this, it doesn't work. Yeah. Okay. Microorganisms that we find in the atmosphere, and especially in clouds, because we are working in clouds, of course they come from the earth. So they are not as a kind of magic thing. They are going for a circle, so they are emitted from the water, from the soil, and from the vegetation, and it's quite important. Vegetation, I will use the word phyllosphere. It's the place where the bacteria live on the leaves of the different trees and plants and so on. So they come into the air because they are aerosolized from different mechanisms. The main one is the wind. Of course, it will take the particles out of the soil, out of the trees, but also by bubbling mechanisms. So, for instance, everybody knows if you go to the ocean, there are vaporization, bubbling out of the water into the air, and then it comes into the dry air. And then there will be particles in the air, they will be dispersing, so in both directions, in vertical, but also horizontal direction. And finally, they will, let's say, it takes a long time to, okay. They will become a cloud because, as any particle of a small size, people of the atmosphere call that CCN, they will condense water at their surface, and so they will become a droplet. So they will become into the cloud because they are themselves droplets. And then they will go down to the soil, mainly by wet deposition, so by precipitation, rain, snow, and so on. Sometimes they can also go down by dry deposition, but for bacteria which are rather small, it's rather the wet deposition which is the main important. So we've been working on different topics, but today I will focus on three main topics. The first one is how they help to make the droplets. Do they have an influence in the formation of droplets? Then when they are in these clouds, do they play a role in cloud chemistry? Are they an alternative to the photochemistry, for instance? And finally, some of these microbes, some of them, not all of them, they can produce ice. And then they can help to the precipitation, because somebody of you know, before having rain, you have ice. And so they can make ice and maybe help to precipitate. Okay, now what are these microbes? So we study microbes in the clouds because we are lucky to live in Clermont-Ferrand, which is close to this mountain, which is called the Puy-de-Dôme. So the Puy-de-Dôme is a volcano. It's 1,465 meters high. It is in the center of France. You have France here, Spain, England. And is often covered with clouds. And on the top of the Puy-de-Dôme, there is an observatory, which is very well known. And now it is labeled as a global atmospheric watch site. So it means it's one of the 30 sites all over the world that's watching the quality of the atmosphere. So there it is a nice place where you have the observatory here, which is fully equipped for, of course, meteorology, looking at all the parameters as you have here on top of the roof. But also there is a laboratory for chemistry and also a laboratory for microbiology, as you can see here. And in addition to that, of course, we have on the roof, we have cloud collectors where we can collect cloud. And so we are working on the cloud water that we collect on top of this station. And for people who are not very familiar with that, this is a droplet impactor. So the principle is quite easy. So you put your cloud impactor facing the wind, and then if it is in the cloud, so it means the cloud has to be on top of the mountain. And then you have an aspirator that make aspiration here, and you have a design, specific design here. We have a slit which is designed in such a way that cloud water will go through this site and the air will go around. So the air will be flushed out. And so we collect the cloud water, and then we can study this cloud water. Of course, we have to work under sterile conditions because we are working with microbiology. So we work, I can go back to the other, maybe here, you can see a picture of Muriel, you know him, Louis. So you have gloves, you have mask, and you are equipped to be, everything has to be sterile, of course, to not contaminate the sample. Okay, and then what kind of bacteria we get? If we look at the numbers, so we have been a survey for 15 years now, and this paper was corresponding to 10 years survey. You have very little number, to 10 to the 4 to 10 to the 6 cells per mil of bacteria. If you make a comparison, you have the same in water that you drink. Drinkable water is the same, so it means it's rather pure. There are not so many bacteria, but still. And there are some spores and some yeast, and you see it's one order lower because they are bigger anyway, so they are in the same range. Now, what kind of bacteria? So for a long time, we have been studying only the culturable bacteria. So I mean, we collect the cloud water, we put the cloud water on petri dishes, and then you culture them, you isolate them, you sequence them, and then you know what type of bacteria you have. And so this is the result for 10 years. Basically, concerning the bacteria, you have main genera, which are Pseudomonas. They belong to gamma proteobacteria, and Sphygmonas, which belong to alpha proteobacteria. But you see there are many, many kinds of bacteria. You can find all types of bacteria that live on Earth. And so we have collecting these bacteria, and now we have a big collection of them. So we have about 1,000 strains in the lab, in our collection, and this is very useful when we want to do experiment in the lab. We can use this bacteria and as model of what exists in the cloud. Now, the story, so that was to describe the microorganism, but the big question for us, because we are interested in the processes, how they can behave, and what they can do in the cloud, it was this first experiment that gave us a very first idea. If you grow the cell, you take the cloud media, you do nothing, you put it at 70 degrees. 70 degrees is average temperature in the summertime at the Puy-de-Dôme, and you leave them, and what you can see is they are growing. So the ATP is going up, and the number of cells is going up. It means you do nothing, and these cells, they grow. So they are able, it means they are alive, and they are able to use substrates, so molecules which are present in the cloud water, and they can use it to survive and to divide. So that was the first experiment, very basic, that gave us the idea to say, okay, but if they are alive, then they can do things in the atmosphere, and they can use compounds which are in the atmosphere. And so maybe they can play a role in atmospheric chemistry, for instance. So now we have confirmation of this activity in clouds, thanks to more recent approach, which is the analysis of the DNA and the RNA in the clouds. So maybe it's complicated for people who are not biologists, but before we were studying bacteria and fungi that were growing on a specific substrate, okay, but this represents only 1% of the whole microorganism. Now we can use metagenomics or metatranscriptomics analysis. It means you take the cloud water, you extract everything which is inside, all the DNA, all the RNA, and you analyze it. You sequence it, and then you analyze. And what you can see, I'm not going into detail, but just to remind you what I said before, the main groups which are there are again proteobacteria. And these are the bacteria we were finding by culture. So they are the same which are very important in clouds when you look at all the bacteria present or only the one that you can culture. But more important, you can see that that time they are really alive in clouds, because if you look at the RNA, it means if they produce RNA, messenger RNA, it means they are active. And what again, you can see they are active, all of them, and in particular, these proteobacteria are active. So it means that this is the first proof that on-site, directly in the cloud, because what you take, the cloud water, you fix it And if there is mRNA, it means they are alive. They are still synthesizing RNA. So that's the proof that it is true directly in the clouds. Before, it was in the lab. This is in the cloud. And this is the first experiment that shows it. And we are really happy about this result because proteobacteria are the main one and the main active, then we can use our model strains, the one that who are isolated as the Pseudomonas, and that you can use it every day because they are good models. Before, we didn't know it was really good models or not. So, role in of this microorganism in cloud chemistry. So, as you have shown before, they are active. It means they have a metabolism. Metabolism means you have enzymes. They are active enzymes, and enzymes are nothing else but biocatalysis. So they can catalyze reactions. And then if you look, for instance, what we have shown here, I will show you two examples. One is to look at the transformation of the organic matter, so all these carbon compounds which are present in cloud water. And the second one in the interaction of oxidants. Oxidants are very important in the chemistry of clouds because they are sources of radicals. So they are basics of the cloud chemistry. And so just to remind you the basic things about cloud chemistry. Here you have a droplet, then you have the CCN, then you have the air around, the gas phase. So there are lots of different reactions taking place. One is, first of all, is a mass transfer, okay? Because some reactions are active in the gas phase and then they can be transferred here. Of course, radical chemistry and particular photochemistry, which is active in the gas, in the liquid phase, but also in the interface. Then the CCN, if you have, for instance, iron or nitrates, they can dissolve in the water phase. Then, of course, you have physical problems like precipitation or evaporation, but there is a big reactivity inside the droplet. And to be very simple and basic, you have organic matter, so lots and lots of organic compounds as a mixture, and you have radicals. And the main radicals in cloud chemistry are OH, which is the major one, in particular during the daytime, and nitrate radical. And you have these oxidant species, like in particular hydrogen peroxide and iron, that are the source of radicals. So, of course, all this is cloud chemistry. But the question was then, do these microbes, because they are active, they have biocatalyzer, can they compete with this radical chemistry? So the main question we tried to answer. So just to give you an example of cloud chemistry, again a basic one. A lot of things happen in the cloud, outside, and there are lots of. The basic thing is you have oxidation to smaller and smaller molecules. At the end, you have, for instance, formic acid, formaldehyde, formic acid, CO2. Just for oxidation pathways. So I take your attention about oxalic acid, formic acid, acetic acid, formaldehyde, for instance. And these compounds, if you are a biologist, they are typically compounds that are part of the metabolism of microbes. So they are typically using these compounds as a metabolite, as substrate. You do the same, actually. You can use formic acid, acetic acid. Acetic acid, if you have lemon, then you eat acetic acid and you transform it. So we do the same. For instance, here you have what we call the C1 metabolism in bacteria. It's a specific metabolism that takes C1 compound like methanol. Here, formaldehyde formates CO2. And there are also other metabolisms which are more complex. But just to say that there is also the same type of oxidation that happens with radical chemistry. So the idea was to compare the two. Another big thing is the oxidation in the clouds. I have a problem to put that. So as I tell you before, hydrogen peroxide and iron are very important in cloud chemistry. I'm not going into detail in all these reactions, which are very complex, but the result is a formation of OH radicals, which are very, very reactive and that transform most of the organic matter. And you see these compounds, if you imagine that the bacteria are within this system, they face a lot of radicals, a lot of hydrogen peroxide, a lot of oxidant. You have also superoxide, oxygen superoxide ion. These are supposed to be very stressful for the microorganism. But if you look from a closer site, again, they are used to this oxidative stress. I mean, any cell, including you, when you breathe oxygen, you are going to produce reactive oxygen species. For instance, here you have the cytoplasmic membrane, so this is inside the cell, this is outside. When you breathe oxygen, this is what you do now, you are going to produce ROS. So you are going to produce OH radical, superoxide radicals, and H2O2. And what happens in your cells, you have a lot, hopefully, if you don't have these things, then you go to the cell death. You're going to destroy things for the proteins, for the DNA, and so on. But hopefully, we have something to protect us, which is called oxidative stress. And then there are different molecules that will stop these radicals. So, for instance, vitamins, pigments in the membranes, there are lots of these bacteria. They are red, orange, red, and there are pigments that stop these radicals. And also you have enzymes that will transform, for instance, superoxide radical, which is very stressful. By this superoxide dismutase, it is an enzyme, it will produce H2O2. And H2O2 will be transformed by another enzyme to oxygen and water. This is okay for the cell. So, as you can see here, the cells are ready to face this oxidative environment. And what happens with clouds? You have iron, you have hydrogen peroxide, you have the light here, and you are going to produce the same type of errors that will transfer inside the cell, but bacteria, they are used to that. So they are going to face that and to transform these molecules. So you can see from one side, on the carbon side, we have enzyme equipment to transform this organic matter. On the other side, from the oxidative stress, we have also enzyme and different molecules that can face the presence of oxidative component. So that was the idea. We wanted to prove that it could be the case with bacteria from the clouds. And we did a lot of experiments concerning cloud chemistry and carbon metabolism. But I will show you just one example. This one was done with real cloud samples, so I mean not bacteria isolated, but just the cloud sample we took at the Puy-de-Dôme station. There were chemical mixture, very complex, and all the biodiversity of the microorganisms. We didn't isolate, there were many thousands of different cells in the cloud. And then so this cloud, we choose 3 different clouds. So the Puy-de-Dôme is here, okay? And here you have back trajectory showing the cloud origin. And we are lucky in Clermont because this super site is considered as a good model to study the anthropization of the cloud, because if the cloud comes from the ocean, for instance, it will be rather unpolluted cloud from the ocean. If it comes from that direction, it will be highly polluted because it goes through different big towns and big regions of industry. And it can come also from the south. It's not very often that it comes from the south. It is in an intermediate situation. So we took 3 different clouds that makes a variety of the different clouds we can collect at the Puy-de-Dôme station. And then we designed this type of experiment, how to compare the mechanisms which are biotic, so due to the microorganism, and due to the sun or to the radical chemistry. So that was easy. We took a cloud sample, so cloud water sample, and then we divided in 2 parts. One, we sterilized this cloud sample. We went through a filter, so there is no more microorganism. So there are only abiotic reactions, chemical reaction. One is in the dark, and one is in the sun. So we use a photobioreactor where you can mimic the sun, which is in the light, in the cloud, and you have the temperature at which the temperature is fitted to one of the cloud we collected. And on the other hand, you have the intact cloud, so there are still all the microbes present here. One is in the dark, so this will reveal the impact of the microbes specifically, while this one is exposed to solar sun. So that, in that case, you have both the bacteria and the sun. And for that, we have monitored different organic acid compounds and also hydrogen peroxide. And the result, I'll just give you the result, is this one. We have measured the rates of transformation of different compounds So, for instance, oxalic acid, formaldehyde, malonate, succinate, acetate, formate. These are the main compounds you can find in cloud water. And in red, you have what is produced or degraded. This is degraded by photochemistry. This is produced by photochemistry. And the blue ones are due to microbial activity. So we can see here, except for oxalate, where microbes, they don't like oxalate, they never degrade it. For the rest, they can easily degrade it. And it means we are in the same range of order. I don't say that microbes are better than photochemistry, but they are the same range of order considering the rates. So it means, okay, we knew that the enzymes were present, okay, but in this type of experiment, we show that they are in the same range of order. So it means it has an interest to look at them because they can really have an impact. Because if they are active but only for 0.01% of the activity, then forget it. But from the lab experiment, with a real cloud, with everything non-cultured, just like that, it has an impact. So that was the first result. The second one was looking at hydrogen peroxide degradation. As I remind you here, it is the main source of OH radicals. And again, we have made the same experiment, and here this is the result during the daytime and nighttime. During the daytime, it means when you have the light on, of course, the major part is due to the OH radical activity. You can see here. But still, there is some microorganism activity, which is not zero. And of course, during the nighttime, you have OH radicals are much lower. You still have nitrate radicals, and you have also all the phantom, photophantom, and so on reactions. And in that case, they are dominating. So again, we are in the same range of activity. So it means that they are competing with the transformation of hydrogen peroxide by the light or by the microbes. So this interaction between hydrogen peroxide and microorganisms was really something new, that nobody really thought about it. So we decided to go deeper in understanding what was happening. And here you have the scheme of the formation of all the radicals and so on with the iron, nitrate. And so we look at the interaction between microorganism and hydrogen peroxide, as we did on this cloud sample, but this time we're going to work with isolate bacteria. And then, because we know it is a stress, we are looking also at the interaction between this molecule and the microorganism, the metabolism of this microorganism. So we designed an experiment in the lab. This time we used microbes that we knew, and we described what we call a microcosm mimicking cloud water condition. So we have used artificial cloud solution. So we have in the lab now a solution that mimics marine composition of the clouds, which are the most often found at the Pudum station. Then we have used bacterial strains isolated from clouds, which are selected as reference strains. They are mainly the Pseudomonas, as we have seen, they are the most important. Then we have used a model of iron complex to, in fact, produce these radicals. And we have used artificial solar light that mimic the light which is in clouds. And so we did this type of experiment, as before, in these photobioreactors, but with model strains, so Pseudomonas strain and Sphygmonas. Remember, they are the two main species we can find. And we did a lot of different experiments with or without cloud, with or without hydrogen peroxide, with or without iron complex, and so on. And these are the results. We follow the degradation of H2O2. And what you can see here is we look at the abiotic conditions. In all cases, in the two first cases, they are not so important. You need really iron and hydrogen peroxide to produce OH radicals. So, okay, if this was expected, you have degradation of H2O2 by the light and iron. But here, you have the H2O2 with the bacteria in different conditions, and you can see it's much more efficient in that case. So, it means that, as we showed it before in the bulk cloud water, we found the same with isolates from the clouds, different strains, and we got the same type of result. They are able to transform H2O2. So that was a confirmation of what you've seen before. And when you measure the rates of degradation, they are the same range of magnitude at the rates we measure in the bulk cloud water. Then, the thing that was interesting, what H2O2 makes on the bacteria. And to assess that, we measure ATP. So I don't know if you know that. It's adenosine triphosphate. It's a key molecule of the metabolism of the cell. So all cells produce ATP, is the kind of energy of the cell. So it's a proxy for all the metabolism of the cell. So when you have a large amount of ATP, you're in a good health. You have a low amount, you are in a bad health, if I can express that. So we look at the ATP concentration of the cells, with and without the presence of hydrogen peroxide. So here it's without hydrogen peroxide. As you see, it doesn't decrease, it even decreases with time. But in the presence of hydrogen peroxide, then it suddenly drops down from 15 to 5 picomolar, and then it recovers. And then if you look carefully at the time scale, when does it recovers? It recovers when you have completely degraded H2O2. It means that during the time they are degrading H2O2, the ATP drops down. And then when it's finished, they go up again. So we look at this different time sampling to see what happens to the cell. When it drops down, does it mean they are dying? So we are looking at the cell number. So this shows that it has a strong impact on the metabolism of the cell. But if you look at the number of cells, so what you take at each time scale, you sample your bacteria, you put them on a petri dish like this, and you count the number of cells. One cell will give one colony. So it will give you the number of cells during the time. And what you can see is if they are growing with and without, it is the same. So they are not dying. They are surviving to this pressure of H2O2. They behave normally when you look at the growth curves. So it means that they are facing this stress and they are even growing. And finally, it means that they are modulating their metabolome. What we call the metabolome is all the metabolic pathway, which are many in the cell. They are reorganizing all the metabolic pathway. They are adapting to the thing. So the metabolism is different, but they are not dying. And when it's finished, then they recover and the ATP rise again. So they survive and they adapt to the concentration of H2O2. Okay, so this maybe I'm not going into detail because maybe it's a bit complex, I don't know. For microbiologists in the audience, what happens really? So first of all, you can see that H2O2 is degrading in H2. We have seen that from catalase. And then what does H2O2 as a stress? It will block the ATP synthase, so the enzyme that produces ATP. This is why ATP is decreasing. Its work is acting on glutathione. Glutathione is a sulfur metabolite that will be oxidized or reduced, and so will maintain the oxidative and reduction equilibrium. And then what it does, it also acts on the DNA repair system. As I explained, when you have radicals, it will act on DNA. DNA, it's very important to be repaired, otherwise you're going to die. And the enzymes, the system of repair, that repair the DNA, is linked to NAD. And it will deplete this NAD, so maybe it's too complex, I don't know. In the end, it means that all these systems are impacting NAD, and NAD, and NADH is a cofactor which is always linked to ATP. So every time you modify this oxido-reduction equilibrium, then you modify the ATP content. And this is why we see the ATP content which is decreasing, because they are repairing the DNA, they are combating the OH by glutathione, they do a lot of things to stop the stress. And for that, as a consequence, it changes the ATP content. And then once finished, they come back. Okay, this was in the lab, but we tried to see if this could be the strong link between ATP and H2O2. This is what we saw in the lab. But is it true in the clouds? How to assess that? We cannot go into the cloud to see it. But what we have done is we have done statistics. We have collected, we had in the lab 70 clouds that we collected, And from the clouds, we had different chemical parameters, microbiological parameters. And so we could make some statistics. For instance, some PCR statistic here. So to understand that, we have to look at the different parameters. So these are different compounds we measure in class, like ammonia, sulfate, nitrate, sodium, potassium. We measure the pH, we measure ATP, and so on, the number of cells, the bacteria, the fungi, the temperature, all of the parameters that we measure in these 37 clouds events. But when you look at the correlation, what is highly correlated is ATP and H2O2. So this is also true if you take the samples that are directly on the clouds, not in the lab. And interestingly, you can see that this correlation is true here. And ATP is not linked to the pH, for instance, or the temperature and the number of cells. It's really linked to H2O2. We made another type of statistical analysis, univariate testing, and then we found a correlation factor which is extremely low. It means that the correlation is really high. So it looks like in the clouds, they can adapt to this H2O2 concentration, and the ATP also varies depending on the cloud conditions. So what do you think what is the importance for atmospheric chemistry? So if we look at the transformation of organic matter, we know that radical chemistry plays a major role. This is known for ages. But cloud microorganisms, we have seen that thanks to the catalase and peroxidases, they can act on H2O2, so they can act on the source of these radicals. On the other hand, we have seen that this H2O2 act on the cloud metabolism, and they deplete the ATP concentration. And finally, cloud microorganisms, they act on organic matter directly thanks to the carbon metabolism. And then, I'm not going to present the results here, but we've done what we call metabolomics approach. So instead of looking just at ATP, we are looking at all the metabolic routes that exist in the cells, and you can see what is modified in the presence of H2O2. And what we have seen is what is modified is carbohydrates, carboxylic acid, lipids, amino acids. And this is quite important. Why? Because carboxylic acid, we know they are important in clouds. Carbohydrates, we know they are also present in clouds. Amino acids, that was recently shown, they represent a higher amount of compounds in clouds. So it means that depending on H2O2, which is varying a lot with the day and night, with the season, with the pollution, you will have a modulation of this system. So on this side and on this side, it's kind of cycle, and it makes the thing very complex, making that there is strong interaction between the carbon metabolism, the oxidative stress metabolism, and radical chemistry. So that was the end of the first time of my talk, that was focused on cloud chemistry. Now we move to microphysics. So microphysics, I will say a few things about the droplet formation and precipitation. So droplet formation, so there are physicists in the audience, so you know that droplets, they form, and they are small, and then they grow. And there are some compounds that can be packed at the surface. And recently it has been shown that, or there's a great hypothesis, that if you have surface active compounds, so surface active, for people who are not familiar, you will say surfactants, that will go to this interface, so they will go here at the interface between the droplets and the air. It may change the formation of these cloud droplets, so it will change the growth of the aerosol particle and increase the CCN formation. So that's a recent paper that has shown that. And so the idea here was to look at biosurfactant, because surfactants can be also produced by microorganisms. They are surface active agents. They are produced by microbes as secondary metabolism, and they are very, very diverse. There are many kinds of structure, but there are some specificities. They are always formed from a hydrophilic head, which is, for instance, a sugar. In that case, you have glycolipids or peptide, and they have a hydrophobic tail, which is always a longer lipids. Okay. So we have glycolipids, lipopeptides, and also polymeric surfactants, which are long chain of sugars. And these biosurfactants are extremely active compared to, for instance, surfactants you can buy that are produced by the chemical industry. They are 2 orders of magnitude higher when you consider the activity. So the idea was to screen our strains that are in the lab from different cloud events, and to see how many of them could produce biosurfactants. So these are the bacteria we have tested here. So they are mainly formed of proteobacteria. As I said before, they are the major groups present in our collections. And these are from the phyllosphere. Remember, they are mainly found on the leaves of grass, of trees, and so on. There are some yeast, Basidiomycota, and actinobacteria also really highly present. So we have tested all these cloud isolate to see if they are able to produce biosurfactants. So how do we proceed? We are going to measure directly the surface tension with this apparatus. Maybe you know it. So what is the principle? You grow the cell, then you collect the supernatant, so just the culture medium, and then you measure surface tension. So you put you put your medium in this needle here, and this needle will form a droplet, which is around 10 micron. So this is why it's called also a hanging droplet method. You have this droplet here. And here we have a system with a camera which is filming every seconds or less the shape of the droplets, how it will evolve with time. And at the beginning, your droplet is spherical. And if you just have water, it will stay like that. But if you have a surface active product, like a biosurfactant, with time, the droplet will change the shape, and at the end, it will drop down. And if you measure the size here and the shape of the droplet, then you can recover and you can measure, you can calculate the surface tension of your solution. And so you can get this kind of curve. So surface tension, for instance, of water will be constant and will be always at that value. So they are measured as millinewton per meter. Then if you have just the culture medium of the cell, then it will vary a little bit because it's not pure water, but you can see it's quite stable. And then when you have bacteria, if they are producing these surfactants, this surface tension will really decrease from 70 down to 30 sometimes. And the kinetic is depending on the different strains, because there is first an equilibrium that takes place. Basically, the different molecules go to the interface, and they go at the interface between the water and the air. So it takes some time, as you can see. It takes quite a long time, actually. And then the final equilibrium takes place. And so the final measurement of the surface tension is taken here. Some go very quickly, some go very slowly. But in the end, they can have the same surface tension. And so you can then have an idea what are the ones which are very active and the other one which are not very active. So for instance here, we have considered that if the value we measure is over 50 millinewton per meter, they are not biosurfactant, okay? If they are in this range, they can be surfactants, but they are also in the range of ULIs, for instance, macromolecules which are in the clouds. But if they are in that range, it is so active that in that case we are pretty sure they are biosurfactants. And now if we look at who is producing these biosurfactants, and again what you can see, they are mainly Gamoproteobacteria. So the bacteria that are mainly Pseudomonas and which are from the phyllosphere. So these bacteria, they seem to be very important because we find them, they are the most abundant and they are the most active when they produce surfactant. Okay, now we try to see if depending on the air masses, is there an impact on the formation of this biosurfactant production. So we have here the back trajectories of the different samples. But what we do in the lab is not, usually we don't take too much into account the back trajectories. We measure the composition of the cloud, so the different ion and cation and pH and oxidant power and so on. And we then classify them depending on if they are polluted, continental, marine, or highly marine. So the main difference is by the pH, like you can see, pollutant is more acidic. You also the sulfate, nitrate, and ammonia, which is much higher in polluted ones. Of course, if you look at highly marine, you will find a lot of sodium, for instance. Okay, so this is the way we classify because it's much more accurate than just the back trajectory, because sometimes they go from the ocean and then they go on the earth and then they go back, and they are a mix of the different things. So what you have seen here, we have classified depending on the different origin of the clouds. And here you can see this plot. You can see the values of the minimum values of the surface tension. So they are almost all in the same range, but one is quite different. It is this one. Okay. Oh no, maybe something wrong. What shall I do now? Stop it, I think. No, it's coming back. Okay. So this one is quite different and comes from the highly marine compounds. And it's really the one that really go over the ocean. We have the maximum of sodium and so on. They are really impacted by the ocean. And if you look at the cells which are present in this marine, highly marine composition, if you look at this orange stuff are gamma proteobacteria. If you remember, these gamma proteobacteria, they are the ones which are very, very active. Mm-hmm. And of course here, they are in a very little number, which is quite logical because they come from the surface of the leaves. And this comes from the ocean, so it's normal that there is almost no proteobacteria. Yeah. And because of that, you can see a great impact. So we have a suggestion here that maybe the phyllosphere could be an important factor for the production of biosurfactant, and then a potential impact on this formation of droplets. So that's the work we are doing now. Now we are looking for this biosurfactant directly in the aerosols using mass spectrometry. And we hope we're going to publish that soon. Okay, so our second aspect of microphysics, and this is well known from Louis, for instance, because he knows as much as I do, is the potential role of some microorganism on the formation of ice nuclei. And this is related to some organisms, so not all the microorganisms have this potential. If you look at bacteria, for instance, they are again bacteria from the phyllosphere. And they are going to form ice as a rather high temperature, I mean less than minus 10 degrees, and you will see later they can start at minus 2 degrees, which is really high if you compare to other ice nuclei, that dust, for instance, that will start at minus 15, for instance. And these bacteria, they are from the phyllosphere, and again, they are belonging mainly to the Pseudomonas group, genera. And these bacteria actually were well known in the past because they are plant pathogens. So they were known for the pathologies of plants. Why? Because some of them are pathogen because they are producing some toxins. And in addition to that, not only the toxin, but they make the plants freeze at a temperature where normally it doesn't freeze. So the plant, I always give this example, you take a salad, you put it in the fridge, when it's too cold, the salad becomes too floppy. So it's much easier for bacteria to enter and cross the cell wall, which is a cellulose, because a plant cell is quite difficult to penetrate. So they make freeze, so they become very floppy, and then they inject the toxin, and then they kill the cell. So it's very good. Okay. So it was not in the past. What was known, it was really the process, and it has been discovered that it is a membrane protein. Oh, sorry about my manipulation of the... It's a membrane protein, so it's the out cell membrane, because they are negative, they are gram-negative, so they have two cell membrane. One is outside, and these proteins are exposed to the outside, okay? And they are rather small ones, considering there are lots of proteins, they are not too big, that they can be arranged in clusters. So there are different clusters, and depending on these clusters, they can be more or less efficient. We know it is only a physical process because it works also with dead cells. For instance, you take some of these Pseudomonas syringae, we freeze dry them, we break them until the membrane protein is still there. Of course, you don't have to destroy the membrane protein, but if it is still there, even the cell is dead, then you still have this activity. Since then, it has been shown also some other biological molecules, big molecules which are containing sugars and different things, they can also induce this process. So it's due to the protein, it's not due to the metabolism of the cell, except that they have to produce the cell. They are not having this cell all the time. They produce them under stress, for instance, under cold condition. And if they are starving, there's not enough phosphorus and things like that. In that case, they produce it. So it's a physical problem. Then if you look in a more, you focus on this protein, so the structure has been shown. So this is the molecular structure of the protein. So it's a barrel, okay? And in fact, it acts as a dimer. To be active, you need two of this barrel, and they are linked here through this, they are interacting through these amino acids. And here it's a view of modeling what happens if you add water molecule to this protein. In a few nanoseconds, what happens is all the molecules of water, which are these red dots here, they align along these molecules because there are some specific sites which are organized in such a way that the structure, the legs here, if you look at the atoms, they are exactly the same as what is in a crystal molecule. So it's a perfect template to induce the crystallization. This is why it is so efficient, much more efficient than a dust, for instance. Okay, so when you have this structure, they can produce very, very quickly this ice at the surface of the cells. Okay, so when we started this work, we knew that these bacteria were present on the phyllosphere. We knew that they could be present in snow and rain, but nobody screened bacteria for that. So we did screening in the lab. We started from the isolate, so at that time we had this number of cells. Now we've done much more, but it was in 1912, yeah. So we make a freezing test, which is very easy. You dilute your number of cells, so you know how many cells you have per tube, and then you put it in a water bath and you decrease the temperature for -3, -2, -3, -4, etc. And you look how it freezes. So now in the lab we have a camera that makes it automatically, but you can do it by hand, it's the same result anyway. So then you have this type of curve where you have here what temperature it starts. So I tell you, it can be very active, even at -3. And then you have the number of isonuclei per cell. So you may have this, here you have the example of 3 different bacteria. They start at the same temperature, but then if you look at the number of isonuclei they form, there are 2 orders of magnitude. So these 2 numbers are important, the temperature and the number of nuclei, of course. So this one is extremely active. And I think it's, at the moment, I think it's the most active that has been ever described, with Pseudomonas that we isolated in clouds. So now we wanted to see, okay, this is from the lab. We have isolated cells, but is it the true in the cloud? So what we have done in that case, we have collected cloud samples, 12 cloud samples all along the year. These are the back trajectories. And the idea was, okay, we have a protein. If this protein is active, it means if we heat it at 90 degrees, you are going to destroy the protein. You are going to destroy the 3D structure. So it will not be active. So what you do, you take your sample, you divide it in two parts. One is warm at 90 degrees, and the other one is not warm. So the one which is not warm will give you the total number of ice nuclei. It can be particles, it can be anything which is in the cloud, but it is the total number. The other one, you will have, in fact, by making the difference, you will have the biological number of clouds. So we don't know if it is bacteria, but we know it is biological and due to the protein which has been destroyed. And then we have measured that, and the conclusion is that 90% of these biological compounds are ice nuclei in the cloud. As you can see here, even at minus 13. Of course, here it starts with non-biological, because you have a lot of dust. And if you were at minus 20, for instance, you have many dust. But in this area, they are due to these biological components. Okay. So the same result was obtained in rain and snow, because there are many people working also on other compounds. Now we wanted to work in a chamber to try to create this cloud in a real cloud. It's not a real cloud, at least you have a cloud with droplets, and it is still artificial. So in Europe you have this facility in Karlsruhe, and it's managed by Otmar Müller. Some of you know him. And so we made a campaign there where we injected this bacteria in the cloud chamber, and in particular this Pseudomonas strain, which was so active. So it works. It is a huge building, as you have seen on the picture, and there is a big chamber. It is quite large, 84 meters, and you can inject here by a nozzle a bacterial suspension. You can control the AOH, which is 95%, and you can control the temperature and go down to minus 20 degrees. Then you can have a lot of instruments looking at that. And then you can make a big vacuum and control the temperature, and then basically you are going to create a cloud. And so for people, sorry for people who are really on this field, but for the others, what you can see here, so it's one of the things you can see. There are many instruments, but one of the things you can see is the diameter of the particles which are formed. So when they are here, and that, so that's microns, okay, in the range of 10 microns, you have the formation of droplets. If you are over here, you have formation of ice crystal. Of course, this is also due to other measurements that confirm it is ice, not only the diameter. And so what you do, you are doing this decrease in pressure. So we inject the cell, we decrease the pressure, and then the clouds form. And what you can see clearly is that, of course, you form droplets, and you form also ice crystal. So it's a demonstration that indeed you are forming ice crystal using these bacteria. So because they are only one which are injected. There is no other particles. So we played a lot with that, but I'm not presenting the result. So it was also the proof that they can act as ice nuclei under conditions which are closer to cloud formation. Okay. So the conclusion of this talk is the first is about cloud chemistry. So we have shown that they may play a role in the biotransformation of organic matter, of carbon compounds. They are interacting with oxidants, and they can play a direct or indirect role. And from cloud microphysics, they can maybe play a role in the cloud droplet formation as biosurfactants, or in precipitation between ice crystals. Of course, all these remain hypotheses. Now we have moved to more and more experiments, because there are not so many people working on that topic, so we don't have enough data. And we start now to go to modeling, especially for organic matter. Now we have a project for a 4-year project with the call of our president, who made a call that was called Make Our Planet Great Again, to face the politics of Trump. So we have invited foreigners to come in the lab, and so there were some few elected people, and one of them is coming from NOAA and has been in the lab now for 4 years to model all this. And finally, for people who are not from the atmospheric science, why is it important to look at clouds, and what is important in clouds on the climate? So this is a kind of view about what is the impact of the solar radiation on one hand, what is the the role here of the clouds in the green as compared to the different effects. As you know, clouds may have a double effect. They can stop the radiation, so in a way they can cool the thing, but they can also stop the radiation in the other way, so they can be also a kind of greenhouse effect. But if we look globally at what happens, clouds are rather cooling than warming. So cooling is the most important effect of clouds. But again, we don't know much about the impact of these clouds because many people know that, it has been published many times. If you look at the different factors that will make a change on the climate and especially on the increase of temperature, some are very well known. You know that CO2, methane, and so on. They are increasing for sure the temperature. These are rather increasing or decreasing. But clouds here and aerosol, there is still a question mark. Even if we know it is decreasing, cooling, there is such an error bar here, large uncertainty, which is due to the fact that we don't know too much what happens in clouds and what is the interaction between cloud and aerosol. And this uncertainty is so high, it is the major uncertainty to predict the climate change. So, of course, we know, all of us, we know that it will going to be warmer, but how much? 2 degrees, 4 degrees? That's the important thing. And this, for the moment, is not solved because clouds are so complicated to understand that there are still a lot of uncertainties. Of course, because I'm working on biology, we say now it's time to integrate microbes also in atmospheric chemistry and models, including with the ice nucleation as well. Okay, and now it's time for the conclusion, to thank all the people who were involved with this work. So Pierre Amato, who is a main researcher in the group, Martin Sanselm, who is really in charge of the microbiology service and all the maintenance of all the microbes and so on. Virginie, who is Vinatier, who is more a chemist, she worked a lot on these oxidants. Isabelle Canet and Monir Traika, expert in mass spectrometry and NMR, because we use a lot of these tools. I didn't present that, but it is part of our things. Pascal Renard is a postdoc who worked on these biosurfactants. Elona is a postdoc working on these things in Karlsruhe. Michael worked a lot on the transformation of organic compounds in the clouds. Muriel Joly worked on ice nucleation, and Nolwenn Virgo, the latest one, worked a lot on the interaction between hydrogen peroxide and the metabolism of the cloud. And of course, we are all in the same lab in this Institute of Chemistry, but we work in very strong collaboration with Laurent de Guillaume, who is in charge of the Puy-de-Dôme station concerning clouds, and he's also very good at atmospheric chemistry, so we couldn't work without him. And we have to thank, of course, our institution, like the Ciencias, because I'm a Ciencias researcher, the university where I work, and the funding from the UNAM, the ministry, and the regional council. And to finish, I will thank you for your attention.
Thank you, Professor Marie Delort. ¿Hay algunas preguntas?
Hello. First, thank you for coming here to give us this excellent presentation. I hope you like our university. I have a couple of questions. From the part of chemistry, it wasn't clear for me what is the origin of the H2O2 in the cloud, the origin.
Okay.
And for the microphysics part, I don't know if you can repeat, like, what is the philosphere?
Okay, I said so. The first one is easier. Philosphere is just a name of the place where microbes. Philo is vegetation in Greek, and philosphere is where habitants are on this sphere, so on these leaves. Okay. So it's just, well, I could say surface of the vegetation, it would mean exactly the same. So it's a location on the philos, so on the plants. For the origin of hydrogen peroxide, there are different ones. There are, first of all, a transfer from the gas phase to the liquid phase, because it's produced in the gas phase. It's an important source of the hydrogen peroxide. The other one is due to the presence of iron, of sulfate, or different reactants that are reacting and giving this H2O2. So there are both origins. One is produced directly in cloud water, that one big part is produced from the gas phase and is transferred in the liquid phase.
Okay, thank you.
And rather, it can also be transferred from the liquid phase to the gas phase, but the main flux is from the gas phase. And you can see it from the models. When you see the modeling, the flux from H2O2 is very, very important. So it's higher than the production intact.
One is chemical and one is physical.
They are both chemical, but it's produced first because many compounds are produced in the atmosphere, in the gas phase first, before being transferred in the water phase.
Okay.
But H2O2 is transferred very easily, so it just transfers to the...
Thank you.
And I didn't say too many things, but the concentration in the water phase is around from 10 to 100 micromolar, if you need a range of order.
Thank you.
¿Más preguntas?
So thank you very much for your presentation. So you show us that you have a very great database of microorganisms that has been isolated for mid-latitude clouds. So we are here in the tropics. So would you expect that we have here a completely different population of microorganisms that can behave completely different to what you have found in Europe or in mid-latitudes, or are they going to be similar?
So I don't know too much because of the cloud, because there are not many data. We have data from La Réunion, which is a small island close to Madagascar that belongs to France. We only did 2 campaigns. So there are some bacteria which are completely different, the other are quite similar. Now, if we look at what has been published in the air, because now there are a few papers that came out from the air, there are differences, of course, depending, for instance, on the influence of, which is important, for instance, in the import of the dust from Sahara, for instance, or from Chinese deserts. They can carry different types of cells. What is important is really if we are really close to the ocean. For instance, Japanese, they have published some papers. They have samples really, the sampling site is close to the sea. Then they have different type of bacteria. Also, what is not so clear from the literature, because there are not enough data, is in our case we sample clouds which are in the free troposphere, so they are not normally influenced from the ground. But of course they are influenced by the region. They are still in Europe, for sure. But many people, they sample close to the ground. So in that case, you don't know if it is, it can be even more local, so there is still. I think what is published is, in all cases, the proteobacteria for some reason are always very present. But for instance, we have Firmicutes, which in case are not so big, and in other cases they have a lot. Actinobacteria, we have quite a lot, some are less than that. The same from our metagenomics data, we have the presence of cyanobacteria and algae. In our water, in our place, they are not so important. Although, for instance, people who have sampled close to the sea, like the Japanese, they have quite a lot. So I think they are more or less the same, but they can be under different proportions. But I think I cannot answer clearly because we don't have enough data.
Thank you for an interesting talk. And it was a surprise for me to look at the metabolism of the bacteria similar to the phyllosphere bacteria.
Yeah.
Because we are working with the phyllosphere bacteria and the formaldehyde and all the stress, the oxidative stress is incredible in the cloud. Was a surprise for me.
I think I think let's just my personal opinion, so there's no proof at all, but I think it's not just a chance that these bacteria are the main one in the clouds, and they are reacting, facing this oxidative stress and light and so on. Because when you think more closely, you look at a leaf, and you see the bacteria on the leaf. They are exposed to the air, they're exposed to the light, they're exposed to oxidant. So they are the one which are really ready to cope with this, compared to bacteria, because initially we thought we had more from the ocean, for instance. But in the ocean, except they are mixed up, they are not exposed in the same way to the condition that looks like, also the change in temperature. Because the thing I didn't explore because I didn't have time, but one of the things which is amazing about clouds and microorganisms is the fact it's not only an extreme place because it's cold, it's oxidative. Okay, they can cope with that. It's because it's changing all the time. You know, a cloud, it can be at 15 degrees and maybe 10 minutes after it will be at minus 10 degrees and go down. And also a leaf, in a way, it's exposed to daylight, to cold, and here in Mexico I've seen that during the day, during the night, you may have a big change. And so I think they are the most. First, it's easier for them to go in the air because they dry on the surface, make the biofilm is drying, and then the wind takes them in the sky, and also the rain. When you see a big storm falling on a leaf, when you imagine the size of a microbe, which is 1 micron, and a big storm, then they are jumping out. It has been filmed that. There was a science this year. When it drops, it goes up. So they are easier going in the atmosphere, and they are, in my opinion, prepared to this life because they are the closest atmosphere. Also concerning the C1 metabolism, as you know, they can form methanol, they can form chloromethane, they can form lots of compounds that can be found in the atmosphere as well. So they are also prepared a lot to be active on C1 metabolism, for instance. Even chloromethane, for instance. We had a project about chloromethane where plants are emitting chloromethane and bacteria on the surface are degrading the chloromethane. Which is
I am right if I talk about the microorganisms in the cloud treat the water, treat the because
you can say that in a way. Yeah. For instance, we are the paper is under review at the moment, but we have worked recently on aromatic compounds, on phenol. So I worked on the bacteria and the degradation of phenol in the clouds. So we have shown by metatranscriptomic and also by looking at isolate how they can degrade phenol, and they degrade it as efficiently as rich radicals and so on. So we say maybe they can depollute, but well. Yeah, they participate to the change on the pollutants for sure. Yeah?
Thank you.
Hi, I see you.
Nice talk. Thank you. I think this is a very relevant topic. It's very important for the atmospheric science. So the bacteria acts like as CCN, right?
Okay, yeah.
And they consume the organic matter already there.
Yeah, yes, of course.
So in gas phase, introduce directly to the liquid phase and transfer probably to new particles because the pressure goes down and just form a new organic matter. So, but also the bacteria produce their own organics, right?
Of course. This is one of the topics of
yes, exactly. So it's very interesting to know if there is some competition between the secondary organic also already emitted by for example, by the mass volume and the bacteria to form the CCN, or some kind of a synergistic effects are between them, and just to create more CCNs to form the clouds. So what do you think?
Of course it could be. The only thing we did, it was just one paper we have made on that topic. It was looking at the transformation of sugars, for instance. And they were forming EPS, so big molecules of long molecules of sugars. And as you know, that EPS have been formed also from phytoplanktons in the ocean. It has been shown they were sources of secondary aerosols. But it's just one paper we made, but in principle, of course. For instance, what we have in another paper we have done, I didn't present that, but we worked on this problem of complexation of iron, which is still unknown in the clouds. Everybody takes oxalate as a proxy of complexation, but we know it doesn't work. It should be some bigger molecules. And we have proposed that maybe siderophore could be produced. There are molecules which are rather big, produced by the bacteria to catch to complex the iron, which is, again, the order of magnitude is about 2 orders complex to oxalate. And now we have seen from metatranscriptonic that there are really siderophore which are expressed in there. Now we should look for them again, but again, they are big molecules. They could act also as CCN when it is evaporated. There are many things they could do. Of course, we didn't explore that, but it is there is a potential. Always I say a potential because we don't know, but of course it is a possibility. Because in that, of course, we examine rather the oxidation, but not the oligomerization step. But it could be. I don't know. It will take 20 years to answer, I think. Thank you.
Thank you very much for the exciting talk. I agree with Irma. One of the things that you showed about the stress and how the reactivity or the ATP things in the bacteria are reduced. Coping and coping with that. So I would like to, if you could talk a little bit more about the polluted situation where the concentrations of the stressors are higher. You mentioned that your measurements were made mostly Pudutom and specifically urban or industrial pollution, but I'm interested in your thoughts about the biomass burning and the fact that even though the proteins are killed, then the biosurfactant aspect may still be active, and how would that play in polluted situations?
Okay. So we didn't do that, but what we did using selected strains, not on clouds, completely on clouds, but on strains, on different. We took 5 different strains and we exposed them to extreme conditions that are present in clouds. For instance, concerning hydrogen peroxide, we went up to millimolar of hydrogen peroxide, and until 1 millimolar, they were still coping with that. Okay. We went we made also osmotic stress, because we know that when there's evaporation, in a way, you're going to concentrate the things. So we played with concentration of sodium chloride as a proxy of the ions, and that in that case some of them were fine, some were not so fine. So if you increase too much the concentration, they may not resist to this stress. And then we also went to a very strong stress. We put them at minus 20 degrees and make cycles, 6 cycles after that. Okay, and they are still resisting. Many of them resist all that. But of course, it's a very strong stress, which is quite...
They're alive, you're saying?
Yeah.
Well, you can have that kind of cycle in convective clouds.
Okay,
you don't have much convective clouds.
This is okay. And also, to conclude what you say, I'm pretty sure that activity will not be the same in all the conditions. I mean, there will be clouds where, first of all, if they are liquid clouds, it's much easier. If they are frozen clouds, anyway, chemistry is not also fantastic. It is frozen, but okay, they can survive, but they are not active, or they are active because it has been shown they can be active in ice, but the rate will be so slow that it has not great importance, I think. Of course, pollution, I don't know. I think they can cope a lot with pollution, because if you look what happens on the Earth, we're working with phenol, for instance. So if you have places where it is highly polluted, they can still cope with that. But maybe the rate of degradation, if you go to depollution, no, it won't be important, because the time is too short. Okay. But I think they can resist to a lot of stress, but then the rate of degradation of the compound can be very dependent on the context and can be more or less competitive compared to but the same for chemistry, because now everybody wonders what happens really in chemistry. What is really at the interface? Because all the models, they take the Henry law to model to do the transfer, and now we say, oh, maybe there are a lot of compounds, organic compounds at the surface, how it changes the activity, what really happening. So there will be still a lot of questions. But I agree with you that there are some extreme events where maybe they can behave differently. But for hydrogen peroxide, hydrogen peroxide and light in the cloud, because in the cloud, as you know, you don't have the 250, okay, we are not in the high stratosphere. These they don't mind too much. They are more demanding about the salt stress and freezing and defreezing. It's not freezing, but freezing, defreezing, these cycles. Yeah.
There is a question by internet from Klaus, and the question is if climate change could affect bacteria and clouds, and if yes, how it affects the marine ecosystem.
Well, this I'm not going to answer. For instance, a very partial answer. So the first one is a bit linked to what you said about the chemicals which are here. Are they going to be affected? I think they can resist to many things. Maybe the rate of degradation will be slower, so they are not so efficient in doing things. But in a way, it's the same with OH radical. If you increase the concentration, then the rates will decrease also. So maybe it will be the same. Then for ice nucleation, when we were in Castro, we did experiment where we injected the bacteria and also sulfate ammonia to make covering the cells. And then you could see that they are not so efficient in making the ice. Okay, because it will change the structure of the protein, and so it's not so efficient. So it can be an indication of pollution. We have made experiments varying the ozone concentration. There is no effect, NO concentration there is no effect, but pH has an effect. So if you have acidic pH, then the ice nucleation is decreased. So it could be related for polluted clouds are much more acidic, and we have found the same thing in rainwater. I didn't present, but we work also on rain. And rain, we have made a survey with 16 different, no more than that, but 50 different rain samples, and there was a correlation with pH decrease. So on that, it can be effective, yeah. But then I don't know for the rest because I have no idea.
Thanks.
My question is a little bit similar to the previous one that you answered on concentration. Since your database is 10 years, I understand, and then you've caught at the Clermont station the Eyjafjallajökull eruption. What did that have any effect on, especially the iron?
You mean what on? Can you repeat?
Yes. At your observatory.
Yeah.
You had the plume from that eruption because it went partly to France and to different parts of England. I know it went by there, so you should have seen it.
Yeah, so people in the Clermont in the observatory, they made they monitored that in the air, but we didn't in the cloud.
Okay.
Because you need to be there at the right time. You need to have clouds. There don't have to be no rain, no snow. It has to be there. It has to last enough. So unfortunately, we don't have any. So people working in the air, okay, they have monitored that and they have seen the influence, especially from Iceland eruption. It was a big event that they could follow. Unfortunately, in our case, we didn't have anything.
In La Réunion?
À La Réunion, we have, but there was no eruption at that time. So you have to consider one thing. Maybe I didn't insist on that. When you take a cloud sample, you have to be there, you have to collect it, then you have to analyze it. So basically, it's more than 1 month's work. Okay, it's not a online monitoring like the rest. So we miss a lot of things because even if Clermont is not far, it's half an hour driving to go to the bottom of the Puy de Dôme, and then you have a small train to go if you want. you need at least 2 people, then you have everything to be ready, then you have the cloud to be there and to last enough time to collect it, and it has not been during the weekend because the weekend maybe the technician is not there, so then maybe it's holiday and the student is on holiday, and you know what I mean, and or it's okay and then it vanishes or it begins to rain so you cannot collect it. So in the end we have more or less 1 event per month at the maximum that you can collect. So this is why we missed some important events, that it was a shame. So La Réunion, maybe we have more because we have a new contract starting from October for 4 years that will be based at La Réunion. So maybe we have more, but again it will be in campaigns, it will be not all the time. We have to move there and to collect, so if it is not the right period, but we have more information, yeah. I think it's another topic we never looked at, but interaction with volcanic things would be very interesting. But we are a small team, so unfortunately we don't cover it. But it's a good, I think it would be a very interesting topic.
Do you know what is the effect of electric activity or thunderstorms on the processes?
So somebody in France looked at that, Cindy Maurice actually, and it didn't work. But they tried to make experiments on ground, producing lightning, and to see if there would be a DNA transfer. Because if you are, I don't know if you are a biologist, no. When you are biology, if you do genetics and you want to transfer gene from one cell to the other, you have to do something with the membrane because it will not go through. So basically, you make an electric charge, difference of potential, to make holes. Well, it's not exactly holes in the membrane, and so the DNA can get in. So this is the way you can do cloning, for instance. So the idea was the same, saying, well, if you produce lightning, then you may change the membrane, and then you have transfer of DNA. So the problem is, I think, it can be true on the soil. So, for instance, when they go in the soil and you have a thunder light, because on soil bacteria are very close one to the other. You know, you are 10 to the 7th and to the 8th cells per gram of soil. So they are so close because for the transfer you have to be really close because the DNA otherwise will go somewhere else. So in the clouds it was a good idea, I think, but it is not. I think it's not feasible. It's not feasible because there will be the droplets are too far one to the other for the DNA to go and enter another. Of course, it's just, we don't know the hypothesis, but okay, they thought about the idea, but okay, they will have another possibility, we don't know. The studies are stopped, they didn't follow about that. The idea is maybe when they go in the atmosphere, because of that, with all the stresses they have, the radicals, this lightning, maybe it will help to produce mutation on the DNA. And then later on the membrane, or it will change something that we cannot visualize, but in the future, when they are back to the soil, or it could be to the ocean, to answer to the other guy, then maybe you are going to change the nature of the ecosystem, because it's not something natural to have traveled in the atmosphere. That's also a great idea, but how to prove it? But it can be the case that you change, when they go to a thunderstorm, then they are back, if they are still alive, they are different. Something has been changed maybe.